Good morning, good afternoon, and good evening. Welcome back to Chapter 16. We just got done talking about biofilms. We talked quite a bit earlier. We said that pretty much any chemical that we find, there is a bacterial species that will eat it. We use this to our advantage. We said that bacteria can be used to clean up chemical spills. This process is called bioremediation. Remediate means to bring back to normal. Bio means using living things to bring things back to normal. Bioremediation. What it generally means is we're going to use a bacterium to clean up a mess we made. Bacteria have great nutritional diversity. Nutritional diversity means they can eat just about anything. One given species can't necessarily eat anything, but there are 100,000 species of bacteria, so there's a species out there that can probably eat something that we want them to eat. A great example of bioremediation is the sewage treatment plant. I'm not sure if you've ever driven past a sewage treatment plant. You ever driven past one of these things, these big circular bins that are all full of rocks, and there's these little sprinklers that circle around dripping water on it? Well, the rocks are just a substrate to give the bacteria something to attach to. If we look at how it works, what's being sprayed over the rocks is not exactly raw sewage, but essentially raw sewage. It doesn't have any solid matter in it. It's all liquid. The solid's pulled off and they do something else with it. But this is all liquid waste, literally from toilets, from the washing machine, from the dishwasher, from the sink. This liquid, this water, dirty, dirty, dirty water, is sprayed over these rocks. Well, we said the rocks are a substrate for bacteria to grow on. As the water very slowly trickles down through all of these rocks, what happens is the bacteria that live on the rocks eat the organic matter. They eat the waste. Again, bacteria have very diverse nutritional capabilities. They can eat lots of things. And so this dirty water is sprayed onto the rocks. The bacteria living on the rocks eat the waste, and much fresher water, much cleaner water, comes out of the outflow pipe. When I was in college, we went to and visited a water treatment plant. It was actually pretty interesting. After the tour, we went to the creek where the sewage treatment plant let out its clean water. We took a sample from the outflow pipe from the sewage treatment plant, and we took a sample from the stream above where the pipe let out, and the water that was being pumped out by the treatment plant was cleaner than the stream that it was being released into. They do a good job. So remember, there are three domains of life. The three domains of life are domain bacteria, domain archaea, and domain eukarya. Well, we finished talking about domain bacteria. Remember, sometimes it's called domain eubacteria. Both names work. Domain bacteria, domain eubacteria, same thing. We finished up talking about domain bacteria, and now we're talking about domain archaea. This is the other group of organisms that are prokaryotes that don't have a nucleus. Years ago, they used to be classified with bacteria. They have been since unclassified, declassified, pulled out of the group with bacteria and given their own group now called archaea. Many of them live in very extreme environments, and therefore archaea are referred to as extremophiles. Phile means to love, so extremophiles, they love extreme environments. What kind of extreme environments? Like extremely salty water. Those are called extreme halophiles. They love very, very salty water, like the Dead Sea, like saltwater evaporation ponds. There are some that are extreme thermophiles. At the end of this, we will talk about ecosystems. Probably the first ecosystem that the planet ever had was hydrothermal vents, superheated water being spewed out at the bottom of the ocean containing sulfur compounds. We talked about there are some bacterial species that are able to take sulfur compounds and break them apart and use the energy in them. Archaea. Archaea can do that. And these are called extreme thermophiles. They live in this superheated water. Superheated means literally above boiling. It can't boil because of the intense pressure at the bottom of the ocean, but it is superheated water. They can be found in acid pools. This is in Nevada, and this is superheated acidic water that's being spewed out of these vents, and all of this color that we see, that color is archaea, prokaryotes that are living within this water, living and not only living but thriving within this very dangerous, very hazardous water. Some archaea are methanogens. Again, what does gen mean? Gen means to make or to create. Methane is what methane refers to. Methane is a flammable gas. It's the flammable part of farts. Some archaea are methanogens. What we see here is a dump, a landfill, and what you may or may not be able to see is the pipe coming out of this dump. What's happening is down deep underneath where there's no oxygen, there are archaea that are consuming the leftover waste. Remember, prokaryotes can eat just about anything. Consuming the leftover waste in the dump, and they're creating methane. Well, that methane is being captured and can actually be used to generate power. There are some sewage treatment plants that will use the methane that they create to make electricity. There are some cattle farms that will ferment manure specifically for the process using archaea, specifically for making methane that they can then burn, and they burn that methane to turn into electricity. These methanogens, like we said, they live in the guts of cattle and deer. They live in landfills. They're found all over. So, if we're going to make the transition from a prokaryote to a eukaryote, what is the difference? Well, there's a lot of differences between prokaryotes and eukaryotes. The first and primary difference between a prokaryote and a eukaryote is that eukaryotes will have these membranous organelles. And when we talk about organelles, I hope that you remember that organelles, like the nucleus, like the rough ER, like the smooth ER, they have membrane wrappers. They're made out of membrane. We don't find membranous organelles in prokaryotes. We do find them in eukaryotes. These membranous organelles are likely infoldings of the cell membrane. Anywhere in my notes you see PM, that stands for plasma membrane. Plasma membrane just means the outer surface of a cell. I usually say cell membrane because it's easier to say than plasma membrane. So PM stands for plasma membrane, and that's just another way of saying cell membrane. Cell membrane is the outer surface, the outer skin of a cell. Mitochondria and chloroplasts were very likely endosymbionts. What that means is if we look at mitochondria and chloroplasts, we find some similarities. The first similarity is that they both have multiple membranes. The mitochondria has two, an outer membrane and a folded inner membrane, and the chloroplast has two outer membranes and a highly folded inner membrane. And those highly folded inner membranes are similar to bacterial membranes. Second, inside mitochondria and chloroplasts both, we have a circular chromosome, and we just talked about it. Bacteria have circular chromosomes. You don't. Eukaryotes don't have circular chromosomes. They have linear chromosomes, those typical X shapes that we're used to thinking about. What likely happened was the mitochondria was once a free-swimming bacterium that got phagocytized, that got eaten, but not consumed. It got pulled into this not yet a eukaryote, and instead of the not yet eukaryote devouring the thing, it allowed it to live. It stayed. Some accident happened. That's likely what it was. An accident happened. A happy accident. And this happy accident was this little bacterium was really good at making energy from food. And because it was really good at making energy from food, it was so good at it that it was actually made more food for the cell that gobbled it up than the cell would have gotten if it had just eaten it itself. And so this happy accident was it stayed. Another happy accident was when the chloroplast, same process, got consumed and accidentally not eaten. And this cool little guy was able to make sugar from sunlight. So, two happy accidents. One, take waste products and make more energy out of it. Two, take sunlight and make food out of it. Endosymbionts. They lived inside of a host cell and worked with it and lived